Hi, my name is Bryce Conway. Today I'm here to present to you my enzyme catalysis reaction lab from Bio 200. Let's jump into it. So, to start off, enzymes are molecules that break down larger molecules by breaking the bonds between them, and they basically speed up reactions by, instead of having the bonds break naturally, the enzymes will go and attack those bonds to try and break them down faster so that the molecules can be digested. Enzymes are important in living organisms mainly for digestive purposes, because as the food that people or that animals ingest goes into their system, they need it broken down fast so they can get the molecules that they need into their system. The role of trypsin in organisms, again, is to break down proteins. And the protein that we tested in our experiment would be azocasein, which is a common milk protein. So that's the one that we tested in our experiment. Again, trypsin is active mainly in like the stomach and like intestinal GI tract and the gut location of the animals, because that's where a lot of like the digestive properties go down and all that kind of stuff happens. So our question for our experiment would be which temperature would affect, or which temperature increases the rate of the reaction between the enzyme trypsin and the protein azocasein. We did a previous experiment to this, and we found out that the optimum temperature is around 50 degrees Celsius, but we couldn't actually confirm that, so we're doing another test. But this time we're going to use 7 different temperatures at 30 degrees Celsius, 37 degrees Celsius, 44 degrees Celsius, 51 degrees Celsius, 58 degrees Celsius, 65 degrees Celsius, and 72 degrees Celsius, and 79 degrees Celsius. Our hypothesis was that as the temperature gets closer and closer to 50 degrees Celsius, the rate of the reaction should continue to increase until the temperature gets too high, in which the enzyme will be denatured. So again, 50 degrees should be around the optimum temperature. Our rationale that we used was basically that trypsin performs the catalyzation of hydrolysis on peptide bonds, and this basically is the creation of small peptides through the breakdown of proteins. Trypsin is found mainly in the pancreas, again along that GI digestive tract that secretes enzymes, and the pancreas is around 38 degrees Celsius, whereas the bacteria in trypsin is found on the surface, and then again it just shows that based on our previous experiment we tested and found the optimum pH would be 50 degrees Celsius, and that's what we were trying to test. For our materials and methods, basically we started out taking the protein azocasein, we mixed it with the reaction buffer, and we put in our enzyme trypsin, and then we incubated each centrifuge tube in the different temperatures. Again, the 7 different temperatures ranging from 30 degrees Celsius to 80 degrees Celsius, 79 degrees Celsius. So we incubated them for about 10 minutes. And then once that was done, we took them out of the incubator, we opened them, and we put in TCA, which stands for trichloroacetic acid. We call it TCA. And what the TCA did was it killed the reaction. So it basically stopped it. So no more trypsin would be breaking down azocasein. And this was basically so that at the same point in time, all the reactions were stopped, so we would get accurate measurements, and the time could be kept a controlled variable. We centrifuged the tubes after we incubated them to try and separate the solid material, the solid the casein that was broken down, because when the enzyme breaks the bonds between azocasein, it's casein is the protein, and then the sulfamylamides or the other molecules, and they're held together by peptide bonds. So when those peptide bonds break, you get the casein and you get the sulfamylamides. So we wanted to separate those components and only get the supernatant, which would be the products of the reaction, so that we can measure the absorbance. The control conditions that we used were the pH buffer of 8. That was the buffer for the reaction, and that was basically because in our manuals it said that the optimum pH for the reaction would be at pH 8, and the other groups tested in their previous experiments, they tested and found that pH 8 would be the optimum pH for the reaction. So therefore, we did use that. Our negative controls again explains up here that when in our experimental groups we used 50 microliters of trypsin along with the 500 microliters of azocasein, the 450 microliters of pH 8 reaction buffer, and the 500 microliters of trichloroacetic acid once we centrifuge them. But for our negative controls, we used deionized water instead of the 50 microliters of trypsin. But again, we needed to put that in to make sure that the volumes stayed the same, so that we would get accurate measurements of each substance and not get high variability within the results. We used 3 replicates for both experimental and non-experimental, just so we could kind of get a collection of data and eventually be able to average the difference between them to get a good kind of numbers. which brings us to the results. So our results, this is our graph here. It shows us a very strict bell curve. As the temperature on the x-axis approaches 50 degrees Celsius, the enzyme reaches its highest reaction, the highest absorbance rate, meaning that the enzyme was in fact reacting the fastest that it possibly could. Again, our standard deviation is shown on this graph as well, and there isn't very much overlap except for between groups 8 and 7. However, that wasn't really significant to our experiment because it was on the kind of towards the outlier regions of temperatures that we tested. It wasn't towards the one that we were focusing on. The results that we took from our experiment, we made t-test results here. All of our t-test results were statistically significant. That means that they were all under 5%, which means that we could in fact kind of confirm that, or we could conclude that 50 degrees in fact was the optimum temperature for the enzyme. So again, all our values were statistically significant. We did have a little bit of variability in our results. We did have a few mix-ups and kind of like the amounts that were going in just based on experimental and human error, or machine and human error that we fixed. We kind of like disconcluded those results, but we did take count of them in our whole tables. We just didn't put them with our averages and our p-value tests because they were outliers.